So just to get things started, right? Personally, I've been super impressed and I'm a huge fan of what Tyra has been able to do with so little. You guys have a very robust clinical pipeline of assets. You went public a few weeks back, so congratulations for that. And with a team of only 15 and in less than 2 or 3 years, right? So that I think is a great role model for many early stage companies that we work with. And I was hoping with that context, if you can, in your own words, share your story with us and also Tyra's as to how last few years have been, what has set you up for and what lies ahead.
Yeah, absolutely. Without taking too much time, I can keep this relatively brief and short, but a little bit of background on myself. Scientist by training. I did my PhD at MIT in the medical engineering, medical physics program. I'm really focused on cancer
What
biology
building was
at
that? Like,
the Cancer Research Center. Can you guys hear me okay?
Yeah.
Okay.
What number was it?
It was the HST program, actually. So it was in
E19.
E19 building overlooking the Broad.
Yes.
And the HST program. And no, look, it was a remarkable opportunity for me to obviously open the world to cancer research. That was my foray. And I moved out of the lab. From there, I went to McKinsey, where I did business strategy consulting in pharma, biotech, and then moved into entrepreneurship. I think it was mentioned in my intro, first with a company called Sienna that was repurposing a cancer tech for aesthetics and derm. But then I really founded Tyra with a small team to obviously get back to my personal passion in oncology, but it was also out of recognition of really some remarkable things that had gone on in the world, but even focused back at the Broad with the DepMap project, with Cancer Atlas, and some of the remarkable pieces of data that have been shared publicly over the last 10, even the last 5 years. Yep. We recognized at Tyra that there was an opportunity to be really focused in targeted oncology, not just on targets that had, that were being validated in the clinic, but also structurally on insights from next generation sequencing and liquid biopsy that were pointing to acquired resistance mutations that were showing up in the clinic, that structurally we could understand, crystallographically and structurally could have been predicted to cause some of the current gen inhibitors to no longer work. And really we had this vision that if we were rapid and efficient and very structure focused in the way that we built drugs, that we could build next generation drugs very effectively, quickly to address that unmet need as it emerged in the clinic. And that led us to our first target at Tyra, which was an FGFR3 selective molecule that would be agnostic to gatekeeper mutations that had been seen with infigratinib and are increasingly seen with the other inhibitors when used in urothelial cancer. And then also a second program in FGFR2, where similarly gatekeeper mutations and another structurally defined mutation, molecular break, was showing up in patients with cholangiocarcinoma when they went on this. So we started out with a very narrow focus, and we really built a company around quickly and efficiently making next generation molecules, not only that were highly targeted and selective, but were also built to avoid liabilities at acquired resistance mutational hotspots that were showing up clinically. So that's as brief as I can get on the introduction.
This is great. Again, I wanted to perhaps focus more or follow up more on your message for early stage companies. We work with a bunch of them, and I think that's a very interesting, exciting segment for us because they're learning as to how to focus, how to leverage data that is already out there. So in your last few years, right? What are the things that you have done particularly to move as fast as you have and cover so much ground with such a lean team? Any insights, any thoughts around that would be very helpful.
Yeah, let me paint it in two areas. One around target selection, how we think about that. which is directly references, I think, what's available in the public domain, and also in some proprietary databases for validation as well. The second piece is what's probably more unique and proprietary to us, which is how we've set up our discovery engine, our SNAP chemistry platform.
Mm-hmm.
But on the first piece, the first thing that I always do when thinking about a new target is I go on the publicly available cancer depth map user interface, which I really see as a gift to the world, and start to look at exploring hotspot mutations, CRISPR and RNA dependency data, as well as a whole slew of other data that group's made available. So one of our scientific advisory members, Bill Hahn, was on the original paper there. And I just think the work that they've done is really incredible, and I think it's made a significant impact in the world for companies seeking to build and create drugs. Obviously, that's one layer, and so going deeper is important, and we often go deeper. And I think Abhishek, the group you've set up, is a great example of ways in which you can take that data and move to next level analyses, and I think that's important. But beyond that initial target validation, the step towards clinical validation and the amount of work involved in really both finding, sequencing, and ultimately validating in multiple years and hundreds of millions of dollars in clinical studies is a giant one. And what's really important is that you're bringing your best molecule to that fight. And so where Tyra really works on a proprietary basis is we have a very unique approach to how we very rapidly and effectively generate next generation molecules. We're very structure heavy, so we do our own crystallography in-house. We do it in a really rapid time frame. We can turn over a crystal structure from when we've synthesized a new compound in as little as 2 to 3 days. We do several of these a week
with the bound structure.
Yeah, and we do it across multiple protein isoforms, and we do it across multiple mutants as well, like acquired resistance mutations. We really understand why drugs are failing, why drugs are working. We do it with our own compounds, we do it with competitor compounds, but we generate a massive body of structural data. So that's very proprietary to us, of course. The techniques we use to move that quickly, the data that we now have stored up from having done this rapidly and iteratively across multiple series of compounds. Then we have a very, we have a great team that does all of our cancer cell assays and cancer biology here in-house. And then we have an in vivo team that does all of our animal models to really rapidly validate. That data set we see as being fundamentally important to informing a medicinal chemist on what the next design cycle should be. And so our goal is always to take a current design cycle and within a week try and get structural data, cell data, and even in vivo data back to the chemist so they can iterate. And we think by doing that and cutting that time frame by which we get data back to our chemists, that we can move faster. So we moved from first molecule synthesized to a development candidate on Tyra 300, getting an FGFR3 selected gatekeeper agnostic in 15 months. And that was for literally the first thing we ever did to having a development candidate. We really broke the sound barrier in timelines. And now we've really taken that approach and focused that in on our next program, which is FGFR2. And then we've got a whole set of programs behind that we're going to take this approach. And it's always moving as quickly as possible to getting patients what we think is going to be a better drug than the current gen that's out there.
Now, this is a great segue for something else that we wanted to move the conversation to. And also I wanted to share with others that I always get a chuckle when I am on a Zoom call with you, because I've never seen any biotech CEO with so many tabs of depth map open on their browser. And that has been a consistent part of my experience with you having a Zoom call. And to me, I mean, clearly you're a big fan of how depth map has helped you, but that's one of the main, one of the very good examples of what we broadly look at public data, publicly available data. And in your experience, like, as you have looked at targets, mutations that are coming up, what kind of role or opportunities that you see around public data? How you mentioned for depth map, are there others? Do you think there are gaps? Do you think there are challenges? Any commentary around that would be very helpful.
Yeah, look, there are gaps. Let me start with another. I mean, the Project Genie database.
Yeah.
ESI CBio portal. I mean, an ever evolving and expanding data set is also, I think, an incredible gift to the world and to cancer research. I think it's very important, and I think that those things create a ton of value, and they're done in a way that's shared publicly and broadly. Those are always going to be a step behind. I mean, I think everyone should be sequenced, and I think everyone should be sequenced sequentially ultimately. And obviously, there's
will be,
but that is what I hope the future holds for the world, because it informs therapeutic intervention and decision making and drug research, better than anything else we've done. And I think there's immense value. So that's obviously where the gap is today. And the onus is a little bit on drug developers like ourselves to, very thoughtfully and creatively and appropriately do companion diagnostic work and cross-validation work, and, work with liquid biopsy type techniques or next generation sequencing techniques and also work with advocacy groups to get more patients, physicians, and payers ultimately approved to sequence where it creates value. So tons of work and gaps there. I see it as our job as drug developers. And I'm very optimistic for the future that with what we have today and with where the puck's going, that we're going to get there.
This is a very interesting comment. And I think, increasingly every day, it seems like, it's a question of when, not if, where, like, you hear initiatives from governments all over the world where they are initiating population genomics projects, right? Very interestingly, I just read a press release with NVIDIA and Thailand government. They have partnered together to do a population level genomics for Thailand, Thai population. And that's one of many examples now that is out there, right? So to me, that's a great sign of things to come. That's the future that we are headed towards. And as hearing you respond, I also wanted to touch upon some really compelling successful stories like Flatiron, right, where they have collected a huge amount of data relevant to oncology, to help biotech life sciences companies like yours. There, like, again, our worldview is that the kind of access issues that such data sets have span a whole spectrum, right? In many cases, you may not even be aware of things that are out there, which is very, very relevant to, for example, Tyra, right? So, any comments around that, like, how data that you have already generated for a very particular subset of indication that you are going after? What are the, again, opportunities and challenges there?
Yeah, I think the opportunities are obvious with more information. I think some of the things we're missing and the correlations that can be drawn, come to light. And we do look at things like Flatiron, and there's other real world databases to run kind of in silico clinical trials with populations we're going to look at and understand data that comes out when it doesn't have full placebo control, to really start to map and say, how confident are we that this is differentially valuable before we go and do the actual placebo controlled test to fully validate it. So those things become really, really valuable and meaningful. But ultimately, as we all know, retrospectively looking back into a database can only do so much. And it is the job of drug developers to not only do purpose-built drug design, but then purpose design clinical studies for validation, and nothing I think will ever replace that prospective work. The retrospective looks and trying to take what drugs are out there and matching them best to patients, I think is really valuable. But I think if there's one thing we learned in the process of designing purpose-built drugs is if you didn't design a drug with that purpose in mind, no chemical matter is out there that will have done the job. I mean, you can try and in-license and repurpose all day long, but the reality is that you're threading very thin needles, and you have to have a full design criteria from the very beginning on what selectivity components and acquired resistance, coverage you need to get to arrive at a molecule that hits all of the marks, and you don't stumble upon that, unfortunately. And so we need purpose-built drugs, and we need the roadmap for the way they need to be built from large data sets that are increasingly becoming available.
Yeah, no, that's true. And one last piece, as we're talking about the data theme, right? The proprietary data, like you mentioned your structural biology platform Snap that you guys have done. I'm sure that must be part of like bunch of other kind of proprietary data that you guys generate routinely to drive your discovery programs. Again, what are the challenges there in terms of having access to that data across different stakeholders or security? Can you comment on that, right, as to how Tyra has dealt with it so far?
Yeah, I mean, we take it very, very seriously, and so continually looking at being at the head of the curve, if you will, on data security, and just making sure that we are controlling internally our data. So when we work with partners, this is obviously a very legal, an important legal piece and data sharing and IT piece. And I think again, there's things that are meant to be shared, and there's times that it's meant to be shared. But of course, any company in a competitive space is going to take their data that's enabling them to build, the potentially next generation products we want to build very seriously, as we do. Yeah.
Todd, I am being told like we have a few minutes to wrap up the session. Then I'll open up for questions from other attendees and participants. So again, I think you did hint about like what you and Tyra sees the future to be, where more and more sequencing data would be available from patients. Where is Tyra headed, and where the largely the biomedical data landscape? How do you see that evolving in the next 3 to 5 year time frame?
Sorry, just to clarify the question, is a question where we see the landscape evolving, or more specifically where we see Tyra evolving within the landscape?
The biomedical data landscape, and where Tyra, where does Tyra fit in?
I think I think sequencing of patients, sequencing of tumors, liquid biopsies, I see these increasing, not decreasing. I think their value have very clearly now been demonstrated across a number of different targets and drugs. I see the bigger players technologically that are trying to improve distribution, not only doing a great job in perfecting the science, but also playing well with drug developers like us. And I think that's a really nice thing to see, a collaborative spirit, and also trying to break down the barriers so that we're not bringing clinical diagnostics, to the FDA in silos, but that we've got solutions that can work across, potentially different vendors that can still enable patient selection. And so there's a lot of important challenges and opportunities within that matrix, but I only see, the data increasing for the benefit of patients. And, I think it's important for all of us to be part of that solution to move the puck forward.